Mahima, I think she may be having a class. Shall we begin?
Yes, sir. Yes, sir.
Okay, so we'll start with our class. Okay, in the yesterday's class, we discussed about vectors as one of the main topic, right? Why vector is important, and regarding the PBR322, it is one of the most important. And who knows, related to PBR322 as a synthetic vector, a vector which is synthesized genetically, or a vector which was constructed genetically, which was constructed with the help of genetic engineering. It will be asked in examination or may not be asked. We can't guarantee. So PBR322 is one of the important vector that we have to remember and we have to study. Okay. Who knows, in future, if you are becoming genetic engineers, you may be playing with PBR322. Okay. And we yesterday in our class we also discussed saying that PBR322 is widely used E. coli cloning vectors. Okay. And nowadays we are using this PBR322 as a vector. Okay, so it is a small circular DNA which is produced from E. coli, and this is a synthetic vector. It is synthesized by Bolivar and Rodriguez, right? Uh, P stands for plasmid and B stands for Bolivar, right? The entire name of Bolivar is Francisco Bolivar Zapata, and regarding the R, it is referring to the Rodriguez, Raymond L. Rodriguez. Okay, and 322 is the number whenever we are synthesizing uh, a vector, constructing or genetically engineering a vector, then number is just given. Okay, and we said that there are different features which are associated with PBR322. Just to review few, we can say that it is PBR322 is having origin of replication. It is having a gene, a segment of gene. Here it is shown as green shaded region, okay, of the circular DNA. Okay, and ori is the origin of replication, where the replication of this uh, vector, this DNA starts. Okay, so replication fork formation starts at ori site. Okay, then we are also seeing selectable markers. Okay, let me just quit this. There are so many messages coming and disturbing us, right? So PBR322 is having selectable markers also. It is having genes like ampicillin resistance gene and tetracycline resistance gene. So we are having two genes which are shaded here of this small circular DNA. One is ampicillin resistance gene, and this gene is producing a protein, and this protein will be making the bacteria, or if this is present in a bacteria, it will be making resistant to ampicillin antibiotics. And we are also having another selectable marker. Selectable here means it will be helping in selection of transformants from non-transformants, recombinants from non-recombinants. I hope you do remember the term transformants and non-transformants, which we had discussed yesterday. So we are having ampicillin resistance gene and tetracycline resistance gene here, right? And these genes are producing protein, and those protein will be making the bacteria which is having this PBR322 resistance to ampicillin and tetracycline. Even if we kill them by using ampicillin and tetracycline uh, antibiotics, they are not going to die easily. And we can even see so many cloning sites. We are having so many restriction enzymes like PST1, PVU1, EcoR1, CLE1, HINT3, BAMH1, CEL1, PVU2. All these are restriction enzymes, and these restriction enzymes are having cloning site at these different regions. Okay, these different regions of the DNA, PBR322 DNA can be cut with these different restriction enzymes. So we can say that restriction sites of different enzymes are available. Moreover, they are cloning site. And all of these, if we look at the different restriction enzymes, then we can say that these restriction enzymes are having one cloning site, right? There are no two cloning site for Hint3. Hint3 is having its restriction cloning site at this position only, right? If we want to cut uh, this DNA with the help of BAM-H1 as restriction endonuclease, then we can cut only at this position. There is no cutting position for BAM-H1 in other locations. So we can say that they are different cloning sites and restriction sites, and most of these are having just one recognition site or cloning site or restriction site. Okay. Moreover, we are also having replication of plasmid, which is here, and it is coding for the proteins which are involved in uh, replication of this plasmid. Okay, at the ori site. So these are the important features of the PBR322. Are you all here in the class? I hope you all are able to remember what we had studied yesterday. Are you here? Yes?
Yes, sir.
Okay, so we'll continue. These are the important features, and we also looked at the different features which are required by a good cloning vector. Cloning vector should carry the DNA, right? So cloning vector that we have seen is plasmid vector PBR322. It is a good cloning vector. Why it can be called as good cloning vector? If we look at some of the features which are required by a good cloning vector, then we have uh, good cloning vectors are usually having origin of replication. They are also having selectable marker, and they also do have let one of you haven't switched off your mic, so we are also having selectable marker. Okay, and good cloning vectors are also having cloning sites or recognition sites or restriction sites, and we are also having good cloning vector which can be multiplying, which can clone themselves, which can amplify themselves in host organisms. So these are the must required features to have a good cloning vector. So cloning vectors are usually helping in carrying the gene of interest, and these cloning vectors should multiply themselves in host organism, right? And those cloning vectors are said to the cloning vectors are said to be good if these different features are present in them. Okay. Now yesterday we also looked at a question saying that why is the PBR322 a good genetically engineered cloning vector? And you are also having the diagrammatic representation from your textbook here. And why this is considered as a good cloning vector, we already had understood why it is good, right? We are saying that it is having lots of good features which are required by good cloning vectors, say like PBR322 is having origin of replication at this site, and it is also having selectable markers like tetracycline resistance gene, ampicillin resistance gene. So these are usually helping in selection of the recombinants from non-recombinants or transformants from non-transformants. So it is a good cloning vector. Moreover, it is having different cloning sites or restriction sites or recognition sites for different enzymes, and all these enzymes are having just one single recognition site, right? BAM-H1 can cut at this position, and there is no other cutting position for restriction enzyme BAM-H1, right? And moreover, this genetically engineered cloning vector PBR322 is able to multiply itself or clone itself in host organisms like E. coli and other cells. Okay, so PBR322 is good genetically engineered cloning vector as it is having origin of replication or ori site, selectable marker, cloning site, and these are able to multiply themselves in host cell. Okay, now to continue with this, we can first discuss about the origin of replication. Though we are saying that PBR322 is having ori site or origin of replication, what exactly is ori site? We all are knowing that replication of DNA is usually starting with replication fork, right? So replication fork is the place where there is start of replication, right? So ori site is the sequence of the DNA This entire circular structure is showing you a DNA structure, okay, representing a DNA structure. So ori site is the sequence of the DNA or sequence of nucleotide where the origin of replication starts. Okay, so replication starts here, and that's why it is called as ori site. Okay, and we can insert the gene of interest or desired gene, and desired gene is also known as alien gene, or it is also known as foreign gene or target DNA, right? So that piece of DNA can be linked at ori site. If we could link the gene of interest or desired gene or alien gene at the ori site, then there is easier cloning of that gene. Okay, as there is replication starting at this point, then if we could link the gene of interest here, then there is surely multiplication of the gene of interest. Okay, or desired gene. Okay, so ori site is mainly responsible for how much number of copy of the plasmid is required. If we require, say, like 5 number of gene of interest which is placed at ori site, then this ori site will undergo 5 times multiplication. So ori site is responsible for controlling the copy number or how many times it should multiply. Okay, which is linked with the gene of interest. Okay, so mainly ori site is responsible for replication of DNA, or it is the origin or site of replication. Okay, this is what you need to keep in your mind. Okay, now if we discuss about the cloning sites or recognition sites or restriction sites, we have seen so many enzymes which can be used to cut the PBR322, this genetically engineered uh, synthetic vector can be cut with different enzymes, right? Now, what should be the characteristic of this cloning vector? Whenever we are looking for good vector, good good cloning vector should have only one recognition site for one enzyme. Okay, or if it is having 2 or 3, it's fine, but it should be having less restriction site or cloning site, so that we can easily link the alien DNA, we can easily link the foreign DNA to the vector if the vector is having very few or preferably single recognition site. So good cloning vector is said to have one restriction site or one cloning site or one uh, recognition site. Okay, if we are having many of the recognition sites, then there is difficulty in joining the gene of interest. Okay, so we can say in case of the alien DNA, we can ligate or we can join the alien DNA at a place in this PBR322, particularly at two of the genes, say like ampicillin resistance gene and tetracycline resistance gene. We can join the alien DNA, and regarding which we'll also look at the uh, mechanism or process how we are joining the gene of interest in the gene of ampicillin resistance or in the gene of tetracycline resistance. Okay, so we can ligate or we can join alien DNA at different recognition sites, particularly at two antibiotic resistance gene in PBR322. In place of, say, like ampicillin resistance, we can use PVU one recognition site as an enzyme which is having recognition site at this position, or PST1 enzyme in order to join alien DNA at this position. Okay, or we can use BAMH1 and SAL1 in order to join alien DNA in the region of tetracycline resistance gene. Okay, now, for example, suppose if we have to join our alien DNA at this position at by using BAMH1, and if we have to join here, then foreign DNA can be joined or ligated with the help of BAMH1 sites, where we are now inactivating the tetracycline resistant gene. But we can join BAMH1. How? So in order to join, we are going to cut the DNA. We'll be discussing about that in the next classes. But what we have to understand here is we are having so many cloning site for different recognition, different restriction enzymes, right? So we already know what is meant by eco R1. Eco R1 is a restriction enzyme, right? And likewise, we have all these different recognition restriction enzymes, okay? And the other important point that we have to remember is BAMH1 is also a restriction endonuclease enzyme, which can cut the PBR322 in the region of tetracycline resistance gene. Okay, if we use BAMH1 as restriction enzyme, it can cut in the region of tetracycline resistance gene, and due to cutting by BAMH1, we can insert alien gene here in the in the recognition site of the BAMH1, and due to insertion of new alien gene in the tetracycline resistant gene, then there is inactivation of the tetracycline resistant gene. This gene will not be functioning and it will not be producing a protein, and that protein will not be making this plasmid DNA containing bacteria to be resistant to the and tetracycline. Okay, so tetracycline character or gene will be inactivated due to insertion of alien gene by uh, cutting the gene at BAMH1 recognition site. Okay, though I was discussing many things regarding the cloning sites or restriction sites or recognition site, what you need to understand here is you have to remember that PBR322 is a good cloning vector. Why? It is having so many recognition sites for different enzymes. Okay, and regarding EcoR1, it is having only one recognition site or cutting site at this position, and there is no other recognition site, right? Moreover, BAMH1 can cut only at this position of the tetracycline resistant gene of PBR322, and hence we can say that a good vector should have single recognition site. That is the important part here. Okay, and you have to understand what is meant by cloning site. Cloning site is the region where we are cutting the PBR322 with recognition restriction enzyme so that we can clone the gene of interest, clone the DNA which is desired at this position. Recognition sites mainly because of restriction enzymes are having their cutting region at this position, or restriction sites they are going to cut at this position only, right? So hope you all are following. Though this is a new concept, you may have got idea, and if you go through these different notes or if you go through your textbook, you may get better understanding. Okay, as this is the first time taught to you, right? That's why you may be finding these difficult, but don't consider as difficult. It's very easy. The main concept is about the characteristic features of your PBR322, which is a good cloning vector. Again, we'll discuss what is meant by selectable marker. Selectable marker, the term selectable means it will be process of selection, right? A process of choosing something, and marker here is helping in identify something, right? So selectable marker is helping in identification of uh, recombinants from non-recombinants. Okay, so we'll discuss further with the help of different statement or concepts given in your textbook. The cloning vector should have selectable marker, right? A good cloning vector, if it is having selectable marker, then it will be helping us to identify uh, what are the recombinants and which are not recombinants. So mostly we are looking for selectable markers, say like genes which are encoding resistance to the antibiotics are usually considered as selectable markers. In case of PBR322, we are having ampicillin resistance gene and tetracycline resistance gene. And these genes or these segment of the DNA are considered as selectable marker. Why? These genes are coding resistance to antibiotics. These genes are protein. These genes will be forming a protein, and those protein will be making the PBR322, which is present in an E. coli, resistance to your ampicillin and tetracycline. Okay, so uh, usually the selectable markers are genes which are coding for resistance to antibiotics. Antibiotics are of different types. Some the antibiotics that we are seeing here are ampicillin and tetracycline. We also have ampicillin, chloramphenicol, uh, so many things, right? Kanamycin and all. Now, normal E. coli cells are not usually carrying the resistance against any antibiotics. If we are having E. coli, normal E. coli, if we consume any of the antibiotics, then E. coli will die in our intestine, right? So normal E. coli are usually not having resistance against any of the antibiotics, but they can become. How? By inserting this PBR322 in normal E. coli, then that normal E. coli will become resistant to ampicillin and tetracycline. As PBR322 is having genes which are coding for ampicillin and proteins which are coding for ampicillin and tetracycline. Okay. Now, what is the main uses of selectable marker? As I have already stated, these are helping in identifying the non-transformants from the transformants. So hence we can identify what are the non-transformants and what are the transformants. And if we require the recombinant or transformants, then we can select them and we can eliminate the non-transformants. Okay, hence there is easier selection of the uh, growing transformants from the petri plates. Okay, so we can simply say that selectable markers are helping in identifying transformants from non-transformants, and what we are required is transformants or recombinants. We are not requiring non-transformants. Okay, are you all here? Yes.
Yes.
So we are discussing something new. That's why some of you may be facing difficulty in understanding. Okay, but don't consider it as difficult. So regarding the selection of the recombinants, how it is done? It is done usually by process of by two main methods. Okay, say like we have inactivation of antibiotic resistance gene. uh, if we insert an alien gene here with the help of BAMH1, then it will inactivate the tetracycline resistance gene, and this gene will not be forming protein which will be making the bacteria resistant to tetracycline. So we can say that we have two methods for selection of recombinants. We require the recombinants, not the non-recombinants. We require the transformants, not the non-transformants. So in order to select recombinants, we have two main methods. One is inactivation of antibiotic resistance gene, and the other one is insertional inactivation. So inactivation of antibiotic resistance gene, as we have studied, we are uh, damaging the tetracycline gene. Suppose say like uh, if we are having this gene without cut by BAMH1 and SAL1, then this gene is normal, right? Tetracycline gene is normal. Suppose if we cut tetracycline resistance gene by BAMH1, then there is inactivation. The gene of tetracycline will be cut in between, which means there is destruction, inactivation, right? So inactivation of antibiotic resistance gene is uh, destroying the tetracycline resistance, or it's not directly destroying due to cutting and insertion of new alien gene or desired gene or gene of interest in between the recognition site of BAMH1. There is inactivation. Now this gene will not be active. This gene will not be forming a protein which is making PBR322 resistance to antibiotics. Okay, and we have insertional inactivation, wherein we are inserting the alien gene and there is inactivation of the gene. Quite similar but not exactly similar. Say regarding the inactivation of gene encoding for antibiotics by insertion of alien DNA. Okay, say suppose here we are having the PBR322 gene, wherein we have the ampicillin resistance gene and tetracycline resistance gene. And at the region of the recognition site or cutting site or restriction site or cloning site of BAM-H1, there is cutting by BAM-H1 uh, restriction enzyme, and there is insertion of a new gene, and that's why now there is inactivation of the tetracycline uh, resistance gene or the gene encoding antibiotics. Okay, so one antibiotic resistance gene, that is tetracycline resistance gene, will be helping in suppose if this is inactivated, then the other gene, ampicillin resistance gene, will be helping in selection of the transformants. Okay, uh, and the other gene, that is tetracycline resistance gene, is inactivated due to insertion of alien DNA, and this brown colored segment shows that due to insertion of alien gene. This is the insertion of alien gene. Okay, and there is cutting by BAMH1. Okay, this is quite though I'm trying to explain, even me, I'm having doubt whether you are able to understand or not. But if you just try to follow, who knows, you may be having some idea, you may get some idea. So regarding the selection of whether we have recombinants or not, then we have to go for so many plating, and it will be a really difficult situation or difficult process. So that's why it is a cumbersome process regarding the selection of recombinants from the non-recombinants. It is a cumbersome process. That means it is a difficult process. Okay, and for which we require plating. Okay, so this gene will be inserted into a bacteria, right? Suppose if this plasmid vector, which is containing the foreign DNA, which had inactivated the tetracycline resistance gene, being taken up by a bacteria, then that is called as transformants. Okay, so here within this bacteria there is the gene which is inserted, okay, at the BAMH1 after cutting with BAMH1 and inactivation of the tetracycline resistance gene. So this is transformants, transformant or recombinant. Regarding the non-recombinant, there is no insertion seen here. There is the same normal uh, uh, PBR322 is shown here, right? That means there is no alien DNA, no foreign DNA, no desired DNA. So what we can say is that in this slide, what we had understood in gist is that we are having ampicillin resistance gene and tetracycline resistance gene, which are going to help us in selecting transformants from non-transformants or recombinants from non-recombinants, and how selection is done with the help of these selectable markers, ampicillin resistance gene and tetracycline resistance gene. Okay, and whenever we are cutting one of the gene encoding antibiotics by any of the restriction enzymes, say suppose if there is cut by BAMH1 at this at its recognition site and insertion of a new gene or desired gene at this place, then there is inactivation of the tetracycline resistance gene, and the other ampicillin resistance gene will be helping in selecting us, will be helping us in selecting the transformants from non-transformants. That is the main concept that you have to understand. Okay, so how does selection of the recombinant is achieved, regarding which we have two main methods, right, regarding which we'll be discussing in the next class. Okay, so far what we had tried to understood is regarding the uh, uh, features of good cloning vector. Okay, why PBR322 is said to be a good cloning vector, that is the most important part. And we said that PBR322 is having origin of replication, selectable markers like ampicillin resistance gene and tetracycline resistance gene, and cloning sites, okay, for different restriction enzymes. And this PBR322 can easily clone themselves, can multiply in host organism. Particularly in genetic engineering, we are using E. coli. So PBR322 can easily multiply in E. coli. So these are the main points that you need to remember. Okay, and in the next class, we'll try to see how we are going to select recombinants from non-recombinants. Okay, I hope you have understood just I hope you have understood this topic. Okay, features required by a good cloning vector, and why is the PBR322 a genetic why it is a good genetically engineered cloning vector. These are the most important part. Otherwise, other points are additional and good for you if you have understood. Okay, I don't think it's good if I take more time. Okay, so we'll discuss in the next class. Okay.
Yes, sir.
Okay, so tomorrow, shall we have class in the afternoon?
Okay, sir.
Because I'm having class. Okay, let's see. We'll discuss. Okay, we'll discuss in the class. Okay, tomorrow. Thank you.
Okay, sir.
Thank
Thank you,
you.
sir.
Thank you. Thank you so much.